Good evening, everyone. I'm Markelle Morphet. I'm a PhD student at the University of Toronto Scarborough under the supervision of Dr. Nick Mandrak, and today I'm going to be talking to you about my project comparing eDNA methods with conventional fish sampling methods in agricultural drains. So agricultural drains are waterways that remove excess water from agricultural land, and they do harbor fish communities often. These waterways sometimes require dredging to maintain the flow of water, and this can harm these fish communities. So they are protected under the Fisheries Act and the Species at Risk Act. So the DFO, the Department of Fisheries and Oceans, classifies drains based on sensitive species present, and among other factors, to determine best maintenance guidelines. So the methods that they use to classify these drains are seining, electrofishing, and dip netting, all of which gear types have inherent physical biases, whether it's related to the size and shape of the fish or related to the mechanics of the gear type itself. They may each represent a different community caught in a drain. So eDNA metabarcoding does not have these same physical biases as conventional gear types, and it can be shown to be more sensitive than electrofishing and seining. So I wanted to compare all of these gear types and their ability to detect sensitive species. So in okay, froze. Oh. Where do I have to point at?
I think sometimes using the computer instead will work better.
Okay.
I think sometimes when there's off figures, it's a little slow.
Okay. So essentially I went with the DFO crew down to southwestern Ontario, so not too far from here, just north of Lake Erie, and did my sampling at the same time or just after they did their conventional gear sampling. I used the peristaltic pump method, and I collected samples downstream and upstream of their sites where they would complete a 40-meter seine haul or electrofishing, and then when there was smaller volumes of water, they would do dip netting. So my results have shown so far that eDNA is able to detect more species than both seining and dip netting. However, electrofishing was able to detect more species than eDNA. One thing to keep in mind here, though, is electrofishing took about 3 to 4 hours per site, and collection and filtration of eDNA or water samples took about 40 minutes per site. So if I spent more time filtering more water, then this result may look different. So essentially this figure depicts, the red shows that where a species was detected by both eDNA and the conventional gear at that drain, and the yellow shows that only eDNA detected a fish at that drain. And then the other colors are the conventional gear detections. So we found that there were 244 eDNA detections and 208 conventional gear detections. This is only looking at presence-absence of these species. And 8 out of 25 species were only detected using eDNA methods. The only species that eDNA was not able to detect was the black-side darter. However, we were able to detect this to the genus. And the only sensitive species that was found in any of the drains using any gear was the Chinook salmon, which was detected only by eDNA in 4 drains. So this detection is important because it would warrant a site-specific review by the DFO and the Conservation Authority, and which would thereby establish appropriate maintenance guidelines for the drain. However, we are thinking there is a possibility of this to be a false positive because of the presence of Chinook salmon samples in the lab at the time of sequencing. So this saying that this is there when it's actually not, it could have conservation ramifications in the form of wasted conservation efforts, essentially. The next steps include running updated data through the bioinformatics pipeline and seeing if we can address these possible false positives. Then integrating environmental conditions of these drains into analyses, because these drains do have quite different physical parameters, especially volumes of water. And then evaluate imperfect detection to determine sufficient sampling effort. So I'd like to thank all of those who helped out and were involved, and I'd like to thank the audience for listening to me today.